Initial material required for curd formation is the pasteurized warm milk and the starter, day-old curd. By adding just a spoonful of curd to milk, how can a whole bowl of milk curdles? Let us know what does milk contain, which allows it to curdle, and what does starter do to the milk. Constituents of milk. Milk is an excellent source of vitamins, minerals, proteins, fats and carbohydrates. The characteristic white color of milk is due to the presence of protein called casein. The carbohydrate present in milk is lactose.
I want you to note down these points. This one. The characteristic white color of milk is due to the presence of protein called casein. Please note down in your classwork book. So the important points
Ma'am, for
you can
science,
write
we
down.
need to use
The
a
heading, can we
paper
please
ruled book, ma'am.
constituents of milk?
Sorry.
Saccharide, which is also called milk sugar. Starter, an old curd. Starter is an old curd or an day-old curd, which contains bacteria called Lactobacillus sp. These bacteria converts milk to curd. In an industrial process of curd formation, these cultures are used in fixed amount. So the industrially produced curd yogurt have same taste in their production. The bacteria used are Lactobacillus delbrueckii subsp. Bulgaricus and Streptococcus salivarius subsp. Thermophilus. Picture represents the bacteria Lactobacillus. Here, the flowchart gives an overview of curd formation. Curd or yogurt is a fermented product of milk. Fermentation is the process where the growth of microorganisms in food can be put to good use. Fermented products are good for health as it contains probiotics and easily digestible nutrients. In the fermentation process, microorganisms convert the complex molecules into simpler form and so these are easily digestible. Picture shows the fermentation of lactose to lactic acid. At room temperature, Lactobacillus bacteria present in milk uses lactose and multiplies itself and keeps on multiplying until the conditions are favorable. It is good to incubate for 8 to 12 hours or overnight incubation and after this it should be freezed to halt the bacterial growth. Lactose present in milk is utilized by bacteria and converts to lactic acid by the enzymatic reaction. In this equation, it shows that lactose, which is
So this is the reason why the curd is being kept for fermentation in warm conditions. It will not be kept in fridge or the freezer, because if there are the climatic conditions are very cool, then the bacteria may not try to multiply itself. So when the conditions are favorable, like it is warm, then the bacteria starts multiplying itself, and then the milk curdles. Let us have a look at what happens actually.
a disaccharide, with the help of lactase converts disaccharide sugar into monosaccharides like glucose and galactose. These monosaccharides are converted to lactic acid by bacterial enzymes. Lactic acid accumulates in milk, increases the acid, hydrogen ions, concentration which decreases the pH of the milk from weak acidic to acidic. Lactic acid acts on the globular proteins, casein, present in the milk and denatures them. This denaturation destroys the tertiary and quaternary structures of proteins and the globular proteins are converted into fibrous proteins, thus giving a thick texture to the milk due to coagulation of the proteins. Coagulated casein curdles into curds and the remaining liquid in milk as whey, light yellow colored liquid. These pictures shows the microscopic view of milk and casein coagulation in it. First picture shows the casein clusters and fat globules in milk entity. In the second picture it shows that Lactic acid causes casein to fall apart and rebind to each other in a loose network that traps water and makes the milk into a semi-solid gel. In the third picture, three clusters of casein proteins are seen. In this flowchart, it shows the fate of milk sugar, lactose to lactic acid and to yogurt or curd. There are many different types of curd or yogurt. Most commercial ones are called yogurt rather than curd. Many flavors are added to yogurt to make them rich in other nutrients or to enhance its flavors. Nutrient content. This is not same in all cases, it varies with the type and source of milk.
I want you to note it down.
Health benefits of taking curd. Prevents vaginal infections in women. Promotes healthy skin and reproductive system. Beneficial in lowering levels of plaque and gingivitis. Provides relief to those suffering from colon cancer. Relieves constipation, diarrhea and inflammatory bowel disease. Boosts immunity and prevents infections and allergies. Helps to lower hypertension and prevents cardiovascular diseases. It is also packed with more amount of calcium and is very beneficial to the bones, and consumption of it daily can strengthen the bones and make the teeth stronger and healthier. Useful in treating insomnia. Products obtained from curd yogurt, ghee, buttermilk, butter, cheese, and paneer. Thanks for watching my video. Enjoy the taste of
I'll just show you when and where the cell is being discovered by whom, and about the cell theory we are going to learn about.
Prior to 1665, most humans were unaware that the microscopic world existed. But that year, Robert Hooke published his groundbreaking Micrographia, a book that revealed this previously unseen and unknown world. Hooke was one of a small handful of scientists to embrace the first microscopes, improve them, and use them to discover nature's hidden details. He designed his own light microscope, which used multiple glass lenses to light and magnify specimens. Under his microscope, Hooke examined a diverse collection of organisms. A gifted illustrator, he drew and explained what he saw. This record of his observations became Micrographia. Some of Hooke's images were so curious and extraordinary that people refused to believe they were real. While observing cork through his microscope, Hooke saw tiny box-like cavities, which he illustrated and described as cells. He had discovered plant cells. Hooke's discovery led to the understanding of cells as the smallest units of life, the foundation of cell theory.
Of cells and results in the release of energy from the incomplete breakdown of ingested food material, and is a form of respiration that takes place in the cytoplasm of cells and results in the release of energy from the incomplete breakdown of ingested food material without the involvement of gaseous oxygen. Anaerobic respiration results in the production of intermediate compounds such as lactic acid or alcohol. Certain bacteria and fungi derive all or most of their energy from anaerobic respiration, and the end products are frequently alcohol and carbon dioxide. The process in this case is called fermentation. Fermentation is therefore a form of anaerobic respiration which results in the formation of alcohol as a respiratory byproduct along with carbon dioxide. However, fermentation formation of other organic compounds such as citric acid, butyric acid, oxalic acid, etc. One of the most widely demonstrated, studied, and commercially exploited fermentation processes is the alcoholic fermentation carried out by yeast cells or Saccharomyces cerevisiae. This chemical equation summarizes yeast fermentation. In this experiment, we'll demonstrate alcoholic fermentation in yeast cells.
I want you to write down this equation, how the yeast fermentation takes place.
For this experiment, we'll need the following. A fermentation setup consisting of a large test tube with a tight-fitting one-hole rubber cork through which is inserted a delivery tube bent at 90 degrees in two places. One end of the delivery tube reaches a few centimeters into the large test tube, while the other end reaches to the bottom of a smaller test tube. A clamp stand, dry baker's yeast powder, glucose powder, lime water, paraffin liquid or cooking oil, boiled and cooled water, beakers, stirring rods. To begin the experiment, prepare 5% glucose solution by dissolving 5 grams of glucose powder in 100 ml of boiled and cooled water in a beaker. Also prepare a small quantity of 10% yeast suspension by adding approximately 5 grams of baker's yeast to 50 ml of boiled and cooled water in a beaker and stirring the contents. Take the glucose solution and the yeast suspension in a 5:1 ratio in the large test tube. So, if you're taking 50 ml of glucose solution, for example, add 10 ml of yeast suspension to it. Add enough liquid paraffin dropwise along the inner walls of the test tube so that the paraffin completely covers the surface of the glucose yeast solution mixture. This addition of oil layer is necessary to create an anaerobic environment in the test tube. Now cover the test tube with a cork containing the delivery tube, making sure that the end of the delivery tube lies way above the level of the oil surface. Now seal the cork using glycerol or grease. Secure the test tube using a clamp stand. Once the tube has been secured, introduce the other end of the delivery tube into the smaller test tube containing lime water, making sure that the end of the tube is immersed well below the level of the lime water. Allow the setup to stand for a few minutes. After some time, you'll begin to notice air bubbles coming out of the end of the delivery tube submerged in the lime water. You'll also start to notice a buildup of a frothy layer on top of the oil layer in the large test tube. After several minutes, the lime water begins to turn cloudy white, suggesting that the gas bubbles coming out from the delivery tube could potentially be carbon dioxide, one of the byproducts of fermentation. At this point, disassemble the setup. First, take the tube containing lime water and add 1% hydrochloric acid dropwise to the tube and shake gently. The cloudy white precipitate dissolves completely, suggesting that the white precipitate is indeed calcium carbonate, which is a product of the reaction between carbon dioxide and lime water, which is calcium hydroxide solution. Calcium carbonate readily dissolves in a mineral acid, in this case hydrochloric acid, to form its corresponding soluble salt, in this case calcium chloride solution. Now take the large test tube and smell the contents. You will find that it has a strong ethanolic smell, suggesting that ethanol is also produced as one of the byproducts during the fermentation of the glucose solution by yeast cells in the suspension. This experiment therefore demonstrates alcoholic fermentation by yeast cells to produce carbon dioxide and ethanol as fermentation byproducts.